Welcome back to the Deep Dive. Today we are attempting to decipher the ultimate instruction manual. We're talking about the code that dictates not just our physical form, but influences, well, almost everything from our intelligence to our susceptibility to disease.
It really does.
We are embarking on a really comprehensive look at what's being called the new genetics, exploring the fundamental blueprint of life, and I think more importantly, how that blueprint is constantly being edited and rewritten by the environment we live in.
That's such a crucial distinction to make right at the start. Our mission today is really to give you, the listener, a thorough understanding of this field, moving far beyond, simple textbook definitions.
Right.
We want to get into the sophistication of these processes, how these tiny, minuscule variations in the code create such vast individuality, and how the world around us, and even the world inside us, shapes our ultimate reality.
Okay, let's unpack this. We have to start with the absolute basics, the bedrock of life, DNA, deoxyribonucleic acid. And it's not just that spiral ladder we all saw in high school biology. It is this incredibly long, complex molecule that holds the entire set of chemical instructions.
The entire set.
And these instructions guide our cells to manufacture every single protein that's required for growth, for maintenance, for survival. It's the master plan for every organism.
It is. And if DNA is the instruction manual, then you could think of the filing system for that manual as the chromosome.
Okay.
These are just the meticulously organized structures where the DNA molecules are stored right inside the nucleus of almost every one of our cells. And to define us as humans, we're characterized by having 46 of these chromosomes structured into 23 distinct pairs.
So, okay, we have the DNA, the structure, and then the chromosomes organizing it. How does the cell actually read this information to build a human body? We know that genes, these specific segments of DNA, are sitting on those chromosomes, and the genes instruct the cell's machinery to make specific proteins. But what's the language? What's the instruction written in?
The language is often described as digital, and it's surprisingly elegant in its simplicity. It relies entirely on the sequencing of just 4 chemical bases. We often just refer to them by their initials: thymine, which is T, adenine, A, guanine, G, and cytosine, C.
The 4 letters.
The 4 letters. And they always pair up in a specific way. T always pairs with A, and G always pairs with C. The specific order, the sequence of these base pairs all along the DNA strand, that is what provides the unique, specific blueprint for making any given protein.
So it's like a 4-letter alphabet that spells out the entirety of human existence.
That's a great way to put it. It really is.
And when we talk about the entire instruction manual, the complete set of genes, what is it, estimated around 20,000 or so?
Roughly, yeah, that's the current estimate.
That provide all the necessary information to make an individual member of our species. That complete package is what we define as the genome. Right?
Precisely, the genome. And it's so important to just appreciate the sheer scale here. That human genome is made up of roughly 3 billion base pairs.
3 billion.
And what's truly fascinating, and this is where we're headed next, is that despite that staggering number of instructions, we all share virtually the same human genome. The individual differences that make you unique are a matter of tiny, tiny variations in those 3 billion base pairs.
Which brings us precisely to the heart of the matter: variation, inheritance, and expression. If, like you said, 99.9% of our DNA is identical, where does that crucial 0.1% of difference come from? And why does it matter so much?
The differences come from variations in specific genes. We use the term allele to describe any variation of a gene or, any of the possible forms in which a gene for a particular trait can occur.
Okay, so think of it like if a gene is a recipe for a protein.
Yes.
An allele would be a slightly different version of that recipe, like adding a different spice.
That's a perfect analogy. Different versions or flavors of the recipe.
So let's talk about those specific types of variation, because this is where the depth really starts. The most common type is something called single nucleotide polymorphisms.
Or SNPs, for short. You'll hear them called SNPs.
Right, SNPs. And they're also called polymorphic variations.
A SNP is essentially a typo in the genetic code. It's when a single base pair, say a T, is replaced by a C at a very specific location. So if you imagine the entire human genome as this massive 3 billion letter book.
A single letter change.
A single letter change. And while these are incredibly common, millions of them occur across the human population. They often occur in what we call non-coding regions or have very subtle effects on how a protein functions.
But then you have a variation that's far more impactful, something called copy number variations, or CNVs.
Oh.
And the sources note that these are arguably much more significant than SNP, so we need to be really clear on the difference.
Absolutely. If an SNP is a typo, a single letter, then a CNV is like duplicating or deleting entire paragraphs, or even whole chapters of that instruction manual. It's a huge structural change. A segment of DNA, which could be thousands or even millions of base pairs long, is just duplicated or deleted in one person compared to another. So instead of one letter changing, you have a massive chunk of code being copied 3, 4, maybe even 5 times.
And the reason CMVs are so functionally significant is that they dramatically change the what's called the gene dosage.
Exactly. That's the key. When you duplicate a section of code, you increase the dosage of all the genes within that segment. And this just throws the complex internal regulation of the cell completely off balance.
Which would have huge downstream effects.
Massive. The sources are very clear on this. Copy number variations significantly affect the likelihood of many inherited conditions, including various aggressive cancers and a wide spectrum of neurological and mental illnesses like autism and schizophrenia. They represent a major structural difference in the blueprint.
It's just incredible to think that we're talking about differences so small on a molecular level.
Yeah.
A single letter changing or a segment being copied. But they manifest as these profound, observable differences in a person. Which leads us right to this core distinction between the code and the result. Genotype versus phenotype.
Okay, so the genotype is the organism's entire genetic inheritance. It's the complete genetic potential. It's the hidden blueprint you receive from your parents, those 3 billion letters of code sitting inside the nucleus of your cells.
Whereas the phenotype is the observable reality.
Yes.
This is everything about an organism that we can measure or observe. Appearance, personality, intelligence, height, behavior, eye color, all of it. The phenotype is the realized potential, which is influenced and shaped by everything outside that core genetic code.
And to understand how the genotype builds the phenotype, we have to look at the paired chromosomes. We have 44 non-sex chromosomes, which are called autosomes. And when we examine a gene pair on one of those autosomes, we can see if the 2 genes are identical. If they are exactly the same in every letter of their code, we say the person is homozygous for that specific trait.
And if the 2 genes in that pair differ in some way, so maybe one is an allele for blue eyes and the other is an allele for brown eyes, then the person is heterozygous for that trait.
Exactly. And that heterozygosity is fundamentally what drives the vast biological and psychological variability that we see in the human population.
And those differing alleles, they interact in predictable ways, which gives us specific patterns of heredity. The simplest one, and probably the most common for complex traits, is additive heredity.
I love this model because it's so intuitive. Additive heredity involves interactions among many genes and alleles where their effects literally pile up or add something to the phenotype.
So it's not one gene beating out another.
No, not at all. It's many genes, each contributing small measurable effects that all sum up to the final trait.
And a classic example the source material gives is height.
Yes, height is perfect. It's not governed by a single tall gene. It's an outcome affected by the contributions of about 100 different genes, each one contributing just a tiny fraction to your final physical stature. So the more tall alleles you inherit from your parents, the taller you are likely to be.
But not all genes are equal players, which brings us to the pattern we all remember from school: dominant recessive heredity. This one introduces a clear hierarchy.
Right. In this pattern, the dominant gene is far more powerful and influential. It can completely dictate the phenotype, often expressing itself fully while completely masking the presence of a recessive gene.
But it's not always 100%, is it?
That's a great point. We have to be careful not to view this as a purely all-or-nothing system. The sources do remind us that while the dominant gene usually controls the expression, the effect of the recessive gene can sometimes still be noticed, maybe under certain circumstances or in a very subtle way.
And this mechanism is why we have the concept of a carrier.
A carrier is someone whose genotype includes a recessive gene for a specific condition or trait, but that gene is unexpressed in their own phenotype. It's hidden.
Because it's being masked by a dominant allele.
Exactly. And crucially, even though the carrier doesn't express the trait themselves, half of their reproductive cells, their gametes, will carry that unexpressed hidden instruction. So they have a 50% chance of passing that gene on to their offspring.
Who might then express it if they get another copy from the other parent.
Precisely.
We also have a very important special case that depends on the sex chromosomes: X-linked inheritance. This is often described as mother-to-son inheritance.
Yes, and this happens when the gene responsible is carried specifically on the X chromosome, and it really just comes down to biological math.
Oh, so.
Well, females have two X chromosomes, XX. So if a female inherits a recessive gene on one of her X chromosomes, the dominant allele on her second X chromosome can often mask it. This makes her a carrier, but usually she's symptom-free.
But males are XY.
Exactly. Males are XY. So if a male inherits that recessive gene on his single X chromosome from his mother, he has no second X to mask the instruction. There's no backup copy, so he will express the trait.
And the classic examples are things like colorblindness or hemophilia.
Yes, X-linked colorblindness is the textbook example. Also, certain types of muscular dystrophy.
That distinction, the presence or absence of that second X, is what determines the whole outcome. It shows how complex the code is, even at this foundational level.
It really does.
And as we start to see that the expression of a gene is not static, that the outcome can be masked or altered or enhanced, it's clear the code is rarely the final word. This leads us perfectly into probably the most fascinating area of modern genetics, the dynamic interplay, environment, cells, and multifactorial traits.
This is where we really break away from the old idea of genetic determinism. Very few traits, if any, are purely genetic. Most are multifactorial traits.
Meaning they're affected by many factors.
Many factors, exactly, both genetic and environmental. And these factors interact dynamically. The environment acts like an external editor. It can enhance, halt, shape, or profoundly alter the expression of the initial gene. And this often results in a phenotype that's markedly different from what the genotype might have predicted.
And the field that studies this external editing process is epigenetics. This is the study of how environmental factors affect genes and genetic expression. It's the mechanism of life experience literally marking your DNA.
That's a fantastic way to phrase it. If the DNA sequence is the hardware, the instruction manual itself, then epigenetics is the software that tells the cell when and where and how often to read those instructions.
So how does it work, mechanistically?
Well, it often involves adding tiny chemical tags, like methyl groups, directly onto the DNA itself. When these tags are added to a gene, they can physically block the cell's machinery from reading it. They can silence the gene.
So it's a biological dimmer switch. It's not about changing the letters, which would be a mutation.
No, the letters stay the same.
It's just about turning the volume up or down on the existing instructions.
Exactly. And the significance of this is immense. Environmental events, circumstances, even your diet can affect if a gene is expressed or silenced. This influences every part of the body and brain, profoundly altering development and function over time.
And the sources specifically emphasize that this epigenetic understanding has opened up new hope for educators and behavioral scientists.
It absolutely has. It suggests that things like learning interventions, stable environments, consistent positive input, these things can physically modify gene expression related to academic and socio-emotional development. The blueprint isn't static, it's pliable.
And speaking of environmental factors, there is an environment inside of us that we often overlook, one that functions as a literal second genome. That is the microbiome.
This concept dramatically broadens our definition of self. The microbiome is this vast community of all the microbes, bacteria, viruses, fungi, and so on, along with all their combined genes living in and on the human body, mostly in the gut, but also on our skin and everywhere else.
The numbers are they're staggering. Our own human genome has about 20,000 genes.
Right.
But the source estimates that the combined genes of the microbes we carry, the microbiome, could total as high as 3 million different genes across thousands of varieties. We are genetically outnumbered by our own tenants.
We are, by a lot. And these tenants are not passive passengers. The microbiome exerts immense influence over our health. We know it influences our immunity, weight regulation, the likelihood of developing certain diseases, and even our moods.
How does it do that? How does it communicate with our body?
Well, these microbes produce thousands of chemicals called metabolites. Many of these metabolites, things like short-chain fatty acids, enter our bloodstream, and they directly communicate with our own immune cells, and crucially, with the brain via the gut-brain axis. In essence, the microbiome has a say, maybe even a veto power, over many of our host functions.
And the ultimate link back to variation is that everyone's microbiome is different.
Completely different. Few microbial communities are the same in everyone. Your microbiome is unique to you. It's shaped by your birth, your diet, every antibiotic you've ever taken. So your personalized microbial universe is dynamically contributing to your phenotype and your health profile, right alongside your inherited 46 chromosomes.
Let's look at how this intricate dance between nature and nurture plays out in concrete terms. The source material provides a couple of classic case studies, starting with alcohol use disorder.
This is a perfect illustration of a multifactorial trait. There are definitely specific alleles involved, genetic factors that can create a strong biological pull or change how the body metabolizes alcohol, which influences the risk of dependency.
I've heard about this, the gene that makes some people flush when they drink.
That's the famous example, yeah. Variations in the aldehyde dehydrogenase gene. Some populations inherit alleles that make drinking alcohol extremely unpleasant. It causes rapid flushing, nausea, headaches, and that effectively protects them from developing alcoholism.
But if you don't have those protective alleles,
Then the path toward dependency is certainly made easier by your genetics, but that's only part of the story. The phenotype, the actual development of the disorder, is shaped by so many external factors. Personality traits, the age of your first exposure, your social context, peer pressure, cultural acceptance. The gene is a susceptibility, not a sentence.
another powerful and I think increasingly prevalent example is nearsightedness or myopia. The sources say this has remarkably high heritability, about 75%. So if your parents are nearsighted, you've likely inherited the genetic disposition.
Yes, the genetic link is very strong, yet the absolute explosion of myopia rates in industrialized societies just demonstrates the powerful role of the environment.
Right, it can't just be genes if the rates are changing that fast.
Exactly. Myopia is heavily influenced by age, limited focus at birth, big changes during puberty when the eye grows rapidly, and then it can decrease in severity in middle age. But the crucial environmental factor is social context.
Meaning screens and homework.
Screens, homework, urbanization, and a lack of sufficient outdoor play. The leading theory is that exposure to outdoor light releases dopamine in the retina, and that dopamine actually inhibits the excessive elongation of the eyeball that causes nearsightedness. So the genes provide the susceptibility, but the lack of sufficient outdoor light pushes the phenotype toward nearsightedness.
It's truly amazing how that combination of genetic potential and environmental exposure shapes the outcome. This whole process of potential turning into reality, it starts right at the beginning of life, with the fertilized egg, the zygote.
The zygote instantly begins duplication and division, a process called mitosis. And this is immediately followed by cell differentiation and specialization. The sequence of early development is fixed: zygote to morula to blastocyst, embryo, fetus, and finally the baby.
And the key to this differentiation lies in those earliest cells, stem cells. These are the first cells. And they're just astonishingly flexible. They're pluripotent, meaning they're able to produce any other cell type in the body.
But through the process of cell differentiation, they gradually lose that flexibility. They get epigenetically programmed to become only one kind of specialized cell, a neuron or a muscle fiber or a liver cell. And the sheer scale of this transformation is incredible. A newborn has about 26 billion cells.
And an adult.
An adult has about 37 trillion cells. And what's miraculous is that every single one of those trillions of specialized cells contains a complete, exact copy of the original zygote's genetic instructions.
And the sources remind us that modern technologies like the gene editing tool CRISPR are giving us the ability to intervene in this process, to potentially reprogram those specialized cells back towards a stem cell state or correct genetic errors in situ.
It's the ultimate promise of genetic medicine for sure, but it carries immense ethical weight with it.
Speaking of natural experiments, the process of twins and multiple births has been historically crucial for helping scientists pry apart the influence of shared genetics, nature, and shared environment nurture. We have monozygotic or MZ twins, which we call identical twins. They come from a single zygote splitting very early in development.
Which means they possess the exact same genotype. They are, for all intents and purposes, clones of each other in terms of their DNA sequence.
And yet, even identical twins confirm the power of epigenetics in the environment.
They absolutely do. While they share the same genotype, slight variations in their phenotype are entirely possible. This can happen due to unequal access to resources or just different positioning in utero. The sources also note that rare event where an incomplete split results in conjoined twins.
Then you have dizygotic or DZ twins, fraternal twins.
Right. These happen when two separate ovas are fertilized by two separate sperm. They are genetically no more similar than any other pair of siblings, sharing on average 50% of their genes.
And they're more common, right?
Yes, dizygotic twins occur about twice as frequently as monozygotic twins. And interestingly, the tendency to release more than one ovum, and thus the incidence of DC twinning, is itself a genetic trait, one that varies significantly based on ethnicity and the age of the mother.
Okay, so now let's shift our focus from the standard 46 chromosomes to examine the significant deviations and differences that can occur, beginning with the determinant pair, the 23rd pair, and sex determination.
Okay, so humans normally possess 46 chromosomes, 44 autosomes, and 2 sex chromosomes. Females are typically XX, while males are XY. The critical difference here lies in the Y chromosome. It's much smaller, but it includes the SRY gene.
Sex determining region Y.
Exactly. And this single gene is the molecular switch that dictates male development.
So this means the sex of the offspring is determined entirely by the father. It just depends on whether his sperm is carrying an X or a Y chromosome when it fertilizes the ovum. A 50-50 chance.
It is, and here we should pause to discuss a technical complexity that often gets overlooked, something called parental imprinting. The source makes a brief but really important reference to this.
Imprinting. So it's not just what gene you get, but who you get it from, mother or father.
Precisely. For a few dozen specific genes in our genome, the cell actually knows the parental origin. In some cases, only the copy from the mother is active, and the copy from the father is epigenetically silenced. In other cases, the reverse is true.
And why does that happen?
It's to ensure the correct dosages of certain critical proteins, particularly those related to fetal growth and development. If the imprinting process goes wrong, if both parental copies are silenced or both are active when they shouldn't be, it can lead to severe developmental disorders. It's a key illustration of how subtle epigenetic regulation controls development.
So moving beyond the standard 46, we have to address what happens when that 23 pair count is uneven. When some gametes, sperm, or ova are produced with more or fewer than 23 chromosomes.
The biological reality here is quite stark. Almost all of the resulting uneven zygotes fail very, very early, often before a woman even knows she's pregnant. They simply cannot follow the instructions required for successful development, which leads to spontaneous abortion.
But some numerical differences do persist. The most common one is Down syndrome, or trisomy 21.
Right. This condition is caused by the presence of an extra copy of chromosome 21, so the individual has 3 copies instead of the standard 2.
Trisomy 21.
Exactly. And the extra genetic material on that small chromosome 21 impacts the dosage of about 300 genes. This leads to a cascade effect, what we call the gene dosage effect, resulting in around 300 distinct characteristics.
Such as?
Well, these characteristics include specific facial features, varying degrees of intellectual disability, and an increased risk for certain medical conditions, like congenital heart defects. And while the sources rightly emphasize that individuals with Down syndrome are unique people with diverse capabilities, it remains a condition defined by specific physical and cognitive challenges that stem from that chromosomal abnormality.
The sources also detail the common numerical differences involving the sex chromosomes, the 23rd pair. There are 4 common patterns of variation that specifically involve the X and Y chromosomes.
Okay, so first up is XXY, or Klinefelter syndrome. This occurs in about 1 in 700 males, and the presence of that extra X interferes with typical male development.
So at puberty.
At puberty, the usual male characteristics often fail to develop. The voice doesn't deepen, and they are almost always sterile. Breast development can also occur, and that extra X can lead to subtle learning disabilities, particularly in language skills. It just shows that extra genetic material is rarely a benign thing.
Next, we have XYY, or Jacob syndrome.
Yes, found in approximately 1 in 1,000 males. These individuals are typically taller than average. And while initially there was some unfounded speculation about aggression, the current research cited in the sources indicates the primary increased risk is for mild intellectual impairment, again, often impacting language acquisition.
And then turning to the female variations, there is XXX, or triple X syndrome.
This affects about 1 in 1,000 females. Physically, they generally appear typical, but that triple dosage of the X chromosome can impair most intellectual skills, though usually just mildly.
And finally, XO, or Turner syndrome. This is where the individual is missing a sex chromosome entirely, having only one X.
Yes, this is less common, occurring in about 1 in 6,000 females. Physically, they are short, they often have a webbed neck, and they lack secondary sex characteristics. So they don't go through puberty, they don't lactate or menstruate. They also experience specific learning disabilities, especially with math and spatial understanding, and may struggle with recognizing emotional cues and facial expressions.
All of these examples just underscore how a simple numerical difference in one small package of DNA can reshape an entire developmental pathway.
It's profound.
And beyond these numerical errors, we have to look at gene abnormalities and their impact, the errors contained within the 46 chromosomes.
Right, so we can distinguish between dominant disorders and recessive ones. If a parent has a dominant disorder, on average, half of their offspring will inherit it.
And what's often overlooked about dominant disorders is when they show up. Most of them begin in adulthood.
That's a fundamental evolutionary principle. If a genetic disorder is dominant and fatal in childhood, the carrier usually dies before they can reproduce.
So the gene is selected out of the population.
Exactly. So the dominant conditions we observe today are typically those that are mild or have variable symptoms, or those that only manifest after the reproductive years have passed. Good examples are Huntington's chorea, Marfan syndrome, and adult-onset Alzheimer's disease.
Recessive disorders, on the other hand, are much more prevalent in the population.
They are because they're silently carried by unaware individuals. They can skip generations, which makes them much harder to track and eliminate.
And common recessive disorders would be things like cystic fibrosis, thalassemia.
And sickle cell disease, which is one of the most studied conditions in the world because of its unique evolutionary history.
We also have the specific category of X-linked conditions, things like hemophilia, which is a failure in blood clotting, and Duchenne muscular dystrophy.
Yes, and both of those follow that mother-to-son inheritance pattern we talked about earlier.
Now let's tie this all back to the environment with the concept of dual effects. The sources highlight that both usual and unusual genes affect every aspect of our health, and these effects only really become clear when they interact with an environmental stressor.
Like a global pandemic.
Exactly. The COVID-19 pandemic offered this tragic, real-world experiment. The sources explicitly state that certain genetic backgrounds altered the severity of the disease. For instance, COVID-19 was much more deadly for people with Down syndrome.
Right, likely due to pre-existing conditions and compromised immune function that result from that extra chromosome 21 dosage.
But conversely, people with a gene for sickle cell anemia were actually afforded some protection against severe COVID.
And this mirrors the famous protection against malaria that carriers of the sickle cell trait possess. That's a classic dual effect.
Cool.
A gene that causes a significant disease, sickle cell anemia, can also confer a survival benefit against an infectious environmental threat like malaria or potentially COVID.
And just being male, XY, was identified as increasing the risk of severe COVID outcomes. Which brings us right back to the sex chromosomes and immune regulation differences between the sexes.
But not all genetic variation is about risk. We have to acknowledge the beneficial additive genes traits that, over millennia, provided a clear evolutionary advantage in specific environments.
A great example is skin color. Dark skin is rich in melanin, and that offers protection against intense UV radiation near the equator. It's crucial for preventing skin cancer and folate breakdown.
But if you move to high latitudes where there's minimal sun, that same dark skin makes it really difficult to synthesize adequate vitamin D.
So the evolution of lighter skin in northern climates provided an additive advantage.
Yes, because it aided vitamin D absorbency, which prevents rickets and other related diseases. Neither skin color is inherently better, they were just advantageous in different environmental contexts.
We see the same principle in height and weight. Being short and carrying extra body weight was historically beneficial in extremely cold climates. It maximizes heat retention.
Right, it minimizes your heat to surface area ratio. But in arid, hot climates where people needed to walk long distances for food or water, longer legs and leaner bodies offered a clear advantage for endurance and for thermal regulation.
And even a psychological trait like anxiety can be beneficial. I mean, high crippling anxiety is detrimental in modern life.
Of course.
But the underlying genetic disposition toward vigilance, caution, and hyper-awareness, that low-level anxiety would have been a massive survival advantage in an unpredictable ancestral environment where predator threats were constant.
A very good point. It's all about context.
Okay, so we've covered the building blocks, the mechanisms of variation, and the profound role of the environment. Let's pivot now to the application of this knowledge. The modern genetic frontiers and ethical implications that have fundamentally changed the way we view health, reproduction, and even our food supply. And this all really starts with the monumental achievement of the Human Genome Project.
The HGP, yes. It was this massive 13-year international effort dedicated to mapping and sequencing the complete human genetic code. It reached its essential completion point around 2001, and it provided the foundational text for all of modern biology.
And the sources reveal two major findings from the HGP that really stunned the scientific community. First, humans have far fewer genes than anyone believed. The initial estimates were around 100,000.
They were. And the actual number is closer to 20,000. That was a huge, huge surprise.
Which led to a crucial insight. If our complexity doesn't come from the sheer number of genes, I mean, we have roughly the same number as a mouse or a tiny worm.
Right. It must come from something else. The HGP shifted the entire paradigm of genetics. We learned that complexity lies not in the number of genes, but in the intricate regulation of those genes.
So software.
The software, the complex software of epigenetics, the non-coding DNA, the vast network of regulatory RNA molecules that control when and how those 20,000 genes are expressed.
And understanding that map has led directly to breakthroughs in reproductive technologies, which inherently raise these profound moral questions. The most established of these is in vitro fertilization, or IVF.
IVF involves the fertilization of ova outside the woman's body, followed by the implantation of the resulting zygote. And it has been an absolute miracle for millions of couples struggling with infertility.
But it does carry some risks.
It does. The sources note that IVF carries measurable, though slight, risks, including a slightly higher incidence of congenital malformations and preterm birth compared to natural conception.
And beyond the medical risks, the technology just opens up a host of ethical quandaries, especially when genetic material is stored long-term or donated by others.
Absolutely. Issues around the ownership of frozen embryos, decisions regarding their disposal, the screening for non-disease traits, these become central debates that vary dramatically across cultures and legal systems.
And one of the most contentious applications of this technology is sex selection. The sources highlight that millions of couples worldwide use various preconception methods to choose the sex of their child.
Right, like selecting sperm based on whether they carry the X or Y chromosome.
And this issue puts individual autonomy directly against societal concerns. Sex selection is illegal in at least 36 countries, often because of fears of demographic imbalance or reinforcing gender bias against females. Yet, it remains explicitly legal in the United States.
Which creates a significant ethical and legal inconsistency. Why is a technology that lets parents choose gender legal here when other forms of genetic selection might be severely restricted? The debate really centers on defining where medical necessity ends and elective, potentially discriminatory consumer choice begins. It's a debate society is far from resolving, as the lines between treating disease and enhancing traits get blurrier and blurrier.
Another major frontier with massive global implications is the GMO debate, genetically modified organisms, particularly in agriculture. And the case of golden rice offers a compelling, almost tragic example of this ethical friction.
It really does. Golden rice was developed by scientists to address a colossal public health failure. Over 250,000 children in parts of Asia and Africa go blind every year, and many of them subsequently die because of severe vitamin A deficiency.
And golden rice was engineered to produce high levels of beta-carotene, which the body then converts to vitamin A, a simple, sustainable dietary solution.
It should have been. But despite the potential to save countless lives and prevent blindness, the source explains that golden rice faced intense resistance. It was prevented from reaching the market for decades in many regions.
And the controversy was fueled by deep fears.
Deep fears, yes. Not just about unknown long-term health consequences, but also about corporate control of the food supply, seed patents held by large companies, and the potential for gene flow, where the modified genes could escape into wild rice strains, altering the natural ecosystem.
So it's a perfect illustration of how genetic innovation immediately becomes a social, economic, and political issue.
It does. The immediate benefit to sick and starving populations is weighed against the precautionary principle, the fear of unknown future consequences related to a fundamental part of life, the food we eat. It highlights that modern genetics is rarely just a matter of lab science, it's a matter of global policy.
Given the rapid pace of this research and the complexity of the ethical decisions involved, the need for informed guidance is just overwhelming, which underscores the crucial role of genetic counseling.
The demand is explosive. The sources indicate there is a far greater need for trained counselors than are currently available. It confirms this is a rapidly growing field with excellent job prospects and salaries. There are an estimated 7,000 professional counselors practicing in at least 28 nations, and they act as this indispensable link between raw genetic data and human life decisions.
But the profession faces some significant challenges and limitations every day. Counselors have to manage patient expectations, especially since genetic data can only reveal risks or susceptibilities.
Right, not a guarantee of future reality. A gene for a condition doesn't mean you absolutely get it, especially for these multifactorial traits.
And another serious limitation is the data itself. A lot of the historical genetic research has been disproportionately focused on populations of Western European descent.
Which means the data is often less reliable or less applicable when you're advising individuals from more diverse global populations. This creates inherent imbalances and gaps in our knowledge.
And we noted earlier that genetic research is proceeding much faster than practical medical care can keep up.
Hmm.
We can identify a mutation today, but the cure or the intervention may be a decade or more away.
And that puts patients and counselors in the incredibly difficult position of knowing a serious risk without having an immediate remedy.
And finally, one of the biggest challenges is the sheer volume of ambiguous findings. When you do a full genomic scan, most of the differences you find are classified as variants of unknown significance, a VUS.
Right, we know the sequence is different, but we have no functional data to link that difference to any specific health outcome. It leaves both the counselor and the patient completely in limbo.
That puts an immense weight on the counselor requirements. They have to have a masterful command of complex biological and statistical material and the ability to communicate those abstract odds and consequences clearly to a layperson who's facing life-changing decisions.
And they also have a profound ethical responsibility regarding what are called incidental findings. They have to ask clients in advance to define the circumstances under which they want to know about other conditions that might be discovered during testing conditions they weren't even looking for.
Like learning about an increased risk for an adult-onset cancer while you're testing a newborn for something else entirely.
Exactly. Navigating that communication boundary is ethically paramount.
And that is the ultimate job of the genetic counselor. Translating 3 billion letters of code and a myriad of probabilities
Right.
into actionable, humane advice. It really summarizes our entire journey today.
It does. We've moved from the four simple bases, TAGC, to the astonishing complexity of multifactorial traits, sex determination, and the critical role of epigenetics. The primary takeaway has to be that your phenotype is not a fixed, predetermined outcome dictated solely by the blueprint you inherited.
It is an ongoing dynamic narrative. Your observable traits and your health profile are constantly being shaped and modified by external and internal factors. Your diet, your stress levels, your social context, the diseases you encounter, and all the subtle environmental signals that are turning your genetic volume up or down.
And perhaps the greatest realization from this deep dive is the introduction of the microbiome, that hidden universe of millions of microbes and 3 million genes that we host. It influences everything from how we metabolize food to the chemicals that regulate our mood.
So, as we conclude this deep dive into the new genetics, here is a final provocative thought for you to carry forward. If every cell in your body contains the original, unique 46 chromosomes you inherited, giving you one instruction set, modern science proves you also carry a second, immensely complex and constantly shifting genome, the microbiome, that is unique to you. The future of health will require us to move beyond simply managing the 46 chromosomes we inherited and focus instead on actively cultivating and curating the vast, influential universe of microbial genes that we carry within us.
A powerful thought to end on. It highlights the future challenge of maintaining our own internal ecological balance. Thank you for joining us for this in-depth look at the new genetics.